Oops, Jack.
One sec. I think we are live. Yeah, we are live.
Excellent. Wonderful. Well, thank you everybody that has come for our 6th talk in this Back from the Brink series. You delightful little brinklets, you. Today we have with us James McCulloch, and he is here to talk to us about how to identify absolutely everything, a topic very, very close to my heart. James is a pan-species lister, that group of intrepid individuals who are interested in recording all nature. They're not twitchers just looking at birds, they're not just butterfliers, they're pushing the forefront of identification and recording. James has seen almost 4,000 species in the UK, 3,949 to be exact, which stings a bit because it's significantly more than I have seen, but I'm not going to hold it against him. In fact, I look up to it. James is currently doing his A-levels and is soon going to be going to university to study biology, and he wants to spread the word about biological recording as well as contribute scientifically through his particular specialism of springtails. I am delighted to introduce James.
Thank you very much for that fantastic introduction. I'll see if I can share my screen, so hopefully it should start to appear. Is that up there? Perfect. Okay, so I've got a massive topic ahead of me today, and I'm also acutely aware that I actually have more slides prepared than I have minutes to talk about them. So I'm actually going to launch straight into it. So I'm going to start off with a bit about me and how I sort of got into recording absolutely everything that I could come across. So I was very lucky when I was younger to be able to join my parents on ecotourism holidays in some really biodiverse places. And growing up surrounded by this fantastic wildlife got me hooked originally. To begin with, it was more of the larger things. So it'd be the mammals and the larger birds that I was able to see without having to hold my super heavy binoculars up to my tiny face. And I would sort of copy my dad as he would write down all the sort of species that he would find in those particular trips, and sort of add that to my world bird life list. This sort of translated into creating a British life list as well, of all the birds that I was able to see in the UK. This was mainly restricted to about Sussex and Surrey, with quite a few interesting species in there as well. This here is an American horned lark, which had somehow made the fantastic transatlantic journey across from North America over to Staines Reservoirs in Surrey, which was quite weird, but still cool anyway. Another thing that my dad got me into was using this website, which is iSpot. And it's where anyone can upload their observations, so photos of anything they've come across in the countryside, and then people can add their identifications if it's unidentified, or you can post it with an identification. Now, I'd sort of post my photos of stuff that I'd seen outside, and people would sort of add their identification, so I was able to learn quite quickly the stuff that I was seeing. But as I sort of gained that knowledge, I'd also add my identifications onto other people's observations, and it was quite fulfilling. From there, I moved on to a website called the Pan Species Listing website. This screenshot is a bit out of date, so I've added 7 species since then, but it's where anybody can make an account and they can add all the numbers of all the taxonomic groups that they've recorded, and sort of it'll calculate your total species number. It's quite difficult to compete on there. There's about nearly 300, if not more than that at the moment, which is quite a lot and really fantastic, including some full-time ecologists. So it's tough going to compete against them, but super fun anyway. From iSpot, I met a really helpful naturalist called Brad Scott, who first got me into bryophytes, because he was running a beginner's sort of introductory course in Sussex, going on about the sort of identification of all the bryophytes I could find in this Sussex woodland that we visited. And it really opened my eyes to the huge diversity of bryophytes that you can find, being the mosses and liverworts. I think there's several hundred species of mosses across the UK. And I think since then, where I saw probably about between 20 and 30 species on that trip, I've now seen about 250 as my knowledge has developed. I'd also join other groups, such as the Westfield Fungus Recording Group, which went out on forays across Surrey and Sussex, try to record all the fungal species in those particular regions at the sites that they visit. And learning from people on those forays who are far more knowledgeable about this than I was really good in developing my knowledge base. And also from Brad, I started to learn about springtails. As he'd gone on one of the Field Studies Council courses, I think either in Malham or Preston Montford, learning about soil mesofauna, which are all the tiny animals that you can find in the soil. If you're not familiar with springtails, they're mostly tiny arthropods. The biggest ones can reach a whopping 6 millimeters, and they can be found in soil, sort of up trees, on the seashore, all these sorts of habitats. And I think at the moment I'm at about 80 springtail species, which is quite a lot considering it's a species, it's a taxonomic group that not very many people focus on at all. And this is my favorite find ever. It was found just about 10 meters from my back door in a rhododendron bush. And this particular springtail is Catiana species A. It's never ever been recorded in the UK before, and it's totally sort of undescribed. No one's given a name to it, and just goes to show what sort of discoveries you can make if you start to learning how to identify absolutely everything. So why would you try to learn how to identify absolutely everything? I'll just talk a bit about this photo because it's one of my favorite insects. It's a wasp that I found last summer in my garden. It's a female Inostemma parasitic wasp. It's absolutely tiny, just a couple of millimeters long at the most. And it lays its eggs in the eggs of other invertebrates where the larvae then develop and then the adults emerge. And often it needs to reach these eggs in quite inaccessible locations. So it needs a long ovipositor. Its abdomen simply isn't large enough for this ovipositor to sort of be housed. So it has to curl around in this sort of horn-like process that arches over the thorax and head called a cornutus. And I think it looks super cool. Firstly, it's easy to make new discoveries. This particular springtail here is Sminthrornis lawrencei, and I found that in my garden a few months ago. My dad had taken a bird box off our silver birch tree and he'd put it on the patio, and he called to me. I was upstairs at the time, and he said, "James, come and have a look at this bird box, see if there's any sort of invertebrates, interesting invertebrates there might be on there." And I saw these tiny black springtails, and I thought, oh, they'll just be Sminthrornis niger, a fairly common springtail that you can see up trees. But some things just weren't quite right. You see those two white spots between the eyes there. They're sort of quite off-white, quite large, not a shape that I'd usually expect for Sminthrornis niger. And the ocelli here, these form part of the primitive eye. They're white, which I've never seen before in any springtails. I'm still not sure at this point whether that's normal for that species, whether it's just an aberration. But I thought maybe it's worth checking this out. Sminthrornis species are quite difficult to identify, but as I had sort of tried to identify them before, I had some experience of what to expect. It's not a great photo, but you can sort of see what I was trying to look at under the microscope here. Springtails are so called because they spring away when they're disturbed, and they do this using the springing organ called the furca, and it's split into 3 parts. The basal part is called the manubrium. The middle part, where the arrow is pointing, is the dens, and the final bit is the mucro. On the dens, at the very tip, on the underside, there's usually either 2, 1, or no setae, so tiny hairs. And in Sminthrornis niger, there should be 1 seta there. In other black springtails like this, there's usually 2, but this one had none. Coupled with the fact that the 5th abdominal segment wasn't fused to the globular abdomen, I realized that this was Sminthrornis lawrencei. And at this point, I was super excited because I'd never seen this species before, and neither have most people on this planet. It's only been seen twice before, not only in the UK, but in the entire world. So again, another exciting find from my garden. Not a remarkable garden, I must add, just a normal, typical garden. Secondly, it allows for greater coverage of all taxonomic groups. Often when you look at a distribution map for a particular species, you'll see that there are clusters of records around certain hotspots. You'll think maybe that species is localized in those particular areas, maybe there's a rare habitat there, but greater monitoring will actually reveal that it's some more widespread species found across the country. But in fact, those clusters of records are just circled around just active recorders, which are going out to their local sites and recording a particular species. If all the naturalists in the UK, which is quite a few, got into recording all taxonomic groups, not just the flashier birds or the moths or the mammals, then we'll have far more coverage for all these taxonomic groups. And the data sort of generated by this will be able to feed into conservation projects much more accurately. Thirdly, rarer species can be casually recognized. Another springtail here, this is Tenothrix atra, and when I first saw this species in Kent, I was so excited. Because this is a species that didn't have very many modern records, so I thought I found a major rarity. I put a photo on Twitter and I was like, oh my goodness, I've just found Tenothrix atra. But then a few days later, I found it in my local nature reserve. And then the next day, I found it in my own garden, and I started to think, maybe it's not actually that rare at all. And I found it a few times since, and I've realized that I was simply overlooking it. And that's probably what most naturalists are doing as well. Because it's so similar to Dichotoma fusca, a species in the same family, that you really do need to look really carefully at features of the antennae to reliably separate it. But since I'd seen that first one, going through my Dichotoma fusca specimens to look for Tenothrix, I realized that it just has a particular look about it that sets it apart. When it walks quite slowly with its antennae sort of moving quite slowly in front of it, with quite distinctive white tips. And it just has a color that's hard to portray in the literature. So it's not something that someone who has never seen this species before would really appreciate. But having gone through all these specimens, trying to identify everything that I'd seen at that site, I sort of got a gist for what I was looking for this species. Now I'm going to go through some general tips on how to identify absolutely everything. In this photo is Wolfia. This is actually the smallest vascular plant in the entire world, so the smallest flowering plant. And this was seen on a Sussex Botanical Recording Society meeting in the Pevensey Levels, which is home to loads of rare aquatic flora. Firstly, make sure to take lots of photos from lots of different angles, because you're not sure what might be needed to identify that particular species. This here is a leaf mine caused by a fly in the genus Chirosia, which is an anthomiid fly. And the fly will lay its eggs between the two epidermises of the leaf, where the larva will hatch, and then it will start to feed on the plant tissue within the two layers of the leaf. And although this one's fairly easy to identify, some aren't. So you may take a photo of a leaf mine and try to identify the species, and then you go home and you realize that actually you needed to see the arrangement of the poo in the leaf mine to identify the species. Because some leaf miners look very similar, they form very similar mines, but the arrangement of poo that they form is different between the two species. Some form more solid lines of poo, some sort of form a string of beads of tiny little poo particles, and that's what's needed to separate the two species. So to avoid any disappointment when you get home, try to take as many photos as you can. For my second tip, pay attention to every detail when comparing photographs. Say when you're comparing to your photo of a winged insect with another photo online of something that you think it might be, make sure to pay attention to every detail. A good place to look with this is the wing venation. So this is the sort of shapes that the veins make in the wings. You can see here that the veins are quite dark in relation to the wing membrane. And in my experience, wing venation is quite invariable within the species, but can vary quite a lot between species. So if you're comparing photos, look at the wing venation, then if there are any discrepancies at all, then there may be a chance that you're looking at something different to what you think it might be. But also check the color of the antennae, color of the legs, color of the abdomen, pretty much anything, to make sure as much matches up as possible. Thirdly, access to microscopes is a handy bonus. So often when you start looking at wasps, for example, or flies, then a lot of the identification work relies on what you can see under a microscope, which can be a bit inconvenient for someone starting out with identifying absolutely everything. In my opinion, it's a really worthwhile long-term investment. The microscope I have now is the one that I first started with, and it's served me brilliantly. But you don't have to own a microscope because there's 60 to 70,000 species of wildlife in the UK, and by no means do all of them require a microscope to identify. Also, if you do need a microscope on certain occasions, then you don't have to buy your own. You can borrow someone's, or you could head to a place like the Angela Marmont Centre in the Natural History Museum, where they have stereo microscopes available for you to loan and to sort of reserve for a particular day or two. Fourthly, target specific species at known sites. This may seem almost counterproductive at first because going to see a species that everybody already knows is already there doesn't really contribute much to data, and so doesn't contribute that much to conservation. But it does get you familiar with that species. So when you're at a different site and you come across this species, then you'll be able to recognize it much more quickly. And those new records will contribute to conservation. Also, meet and communicate with experts. So this photo was taken on the Sussex Botanical Recording Society meeting at the Pevensey levels, where that Wolfia was seen. And it's just one of many examples of days where I've learned so much from people who know much more than I do about biodiversity. Because going out with these people and learning from them as they identify everything that they come across really does shove a load of information in your brain just in one short space of time. And you go away sort of being able to identify a lot more than you first arrived. But you don't have to meet them in person, especially in times like these, but also on the internet. A lot of experts are so happy to identify your photos that you send them as well. Finally, scale. Scale is really important in a lot of instances, because two species which may seem similar may differ on their size ranges. So if you can, take a photo with a scale like a penny or a ruler where it's appropriate. So if you're trying to photograph a flighty butterfly, don't just toss a penny at it, as that might be counterproductive. But if you can, then do. So this is an example of a key which you may come across when you're sort of starting out with identification. This one's quite a complicated one, and it's quite a long one, almost comically long, because it's keying out a family of wasps called the Pteramalidae, which is so speciose, but it's just an example on what you might expect. So you can see you start at the first couplet here, with couplets made up of two different options. This is for a winged insect, so you will look at your specimen and ask yourself whether it's fully winged, so the wings are fully developed as you'd expect, or if they're brachypterous, so they have underdeveloped wings. If the wings are totally developed, fully winged, then we move on to the second couplet, which you can see here, with a number in parentheses indicating the couplet that you came from. So if you look at these two options and you find that neither of them really fit, for example, this is asking for the number of antennal segments, either 13 or 12 or fewer, and yours has 14 segments, you might go back to the first couplet indicated in these parentheses here and sort of reassess the options and perhaps go down the different route. In the second couplet, say your antenna has 11 segments and you move on to the 269th couplet. So this is such a long key, so don't be frightened by this because most are much shorter, but it's just an example. You may also come across keys like these. Now these are rather more confusing. So say you're at the second couplet here. Here you have the number 2. And it reads metanotum without posterolateral macroseti. But where is the other option? Here you have to look at the number in parentheses, and it's not the number that you came from, but rather the number of the line that's the other option in the couplet. Here it says metanotum with posterolateral macroseti. So you'll have to judge that character based on these two lines here, rather than two lines that are sort of right next to each other. And once you've sort of said that your specimen has no posterolateral macroseti on the metanotum, for example, then we move to the option directly below it, which is this third line here, which corresponds to the fourth line as well. So again, I don't expect you to know all of the jargon used here, but it's just an example. There are some good general online identification resources that cost absolutely nothing. One of my favorites is Mike's Insect Keys. This is a really good site because it includes so many keys, either adapted from other works, maybe translated from German or Italian into English in a quite easy to use format. The Royal Entomological Society out-of-print handbooks are really good as well. They're all the handbooks that society has published, gone out of print, and then uploaded as PDFs onto the website. They include lots of keys for flies, for beetles, lots of groups like that. Also NatureSpot, that's a good website that has galleries showing examples of a lot of the, well, all of the taxonomy groups in the UK. It's based around Leicestershire and Rutland, where users there upload their photos for particular species, but it's fairly representative of what you'll find in the UK. So you can scroll through those galleries, try to find a rough match, and then see something that fits, and then look at some specific literature to try to narrow it down from there. Also Flickr, users like Stephen Falk and Brian Evesham have fantastically informative galleries there, which include a lot of bees and hoverflies in the case of Stephen Falk, but also elders and snails and stuff like that from Brian Evesham. And social media as well. A lot of experts can be contacted on Twitter, for example, and as I said earlier, they're usually happy to help. And so now I'm going to go through as many of the taxonomic groups as I can in the time that I have left, which is going to be a challenge, but I'll try to get through it all. And I'll start with lichens. Lichens, if you're not familiar with them, are an organism formed by the symbiosis between an alga and a fungus, sort of working together to form one organism, which I think is super cool. And what helps with the identification of lichens is the substrate that they're growing on, usually rock, usually wood, but sometimes soil. With the rock, you're looking at whether it's acidic or basic, and most people can't just look at a rock and say that's an acidic rock. So what you do is you get a drop of lemon juice, put it on the rock, and if it fizzes, it's basic, because the citric acid within the lemon juice will react with the basic compounds in the rock and cause fizzing, which you'll see there. And also the type of wood is important if it's growing on wood. So specific species of trees will also play host to different lichen communities, because different species have different pHs, so different acidities. Secondly, you can use chemical spot tests. There are two main ones. There's the C test and the K test. The C test is where you add a drop of calcium hypochlorite or some other metal hypochlorite onto the lichen, and that can often produce a color. Usually lichenologists would use household bleach for this. Nowadays, household bleach has other additives like potassium hydroxide, which is an issue. And so if you look through the ingredients list of your bleach and it has potassium hydroxide, you should probably look for another one, because the K test uses potassium hydroxide, and that often invokes a different color change. This potassium hydroxide can be bought in a sort of a 10% or 5% concentration value from various suppliers. I think Watkins and Doncaster sell that, or other microscope suppliers, because it's often used in microscopy as well. You can see in the bottom right there a KC test, and that's just the addition of K, so potassium hydroxide, followed by sodium hypochlorite. You've also got CK test, which is the same thing but the other way around, and that can often invoke further color changes different from just C or K isolated. Now some suggestions on literature. I mainly use the book on the left, Lichens by Frank Dobson, which is good. I think it's recently been updated, a new edition in the last couple of years or so. It's fairly comprehensive with quite a few species listed in there. There's so many in UK, well over 1,000, I think. So it's difficult to include full coverage in just one book, but this does it fairly well to as great an extent as it can. But for beginners, there's quite a lot of fold-out keys published by the FSC, which split lichens into particular habitats or substrates. So this one on the right here focuses on urban lichens on trees and wood, and you can get others for different habitats and substrates. Now moving on to fungi. A lot of fungi will look the same when you're taking a photo from above, so you have to take photos from other angles as well. For instance, characters of the gills on the underside of the cap are often important, either the color or where they attach to the stipe, which is a fancy word for the stem of the mushroom. Either the gills will sort of run up the stem a bit before attaching, or they'll attach perpendicularly, or they'll sort of run down and form an acute angle here when they attach. And the characters of the stem as well can often be quite important. Some genera or families have a ring around the stem. And with some mushrooms, like the mycenas, which are small mushrooms, colloquially known as bonnets, if you snap the stem, then a particularly coloured liquid will be exuded. So in the case of the milking bonnet, it'll be a milk white liquid produced. In the case of the bleeding bonnet, it'll be a more blood red colour, which is also rather cool. Secondly, look at the spores if you can. So you can produce a spore print by cutting off the stem, putting the cap of the mushroom bottom down onto a microscope slide or a piece of paper or some other similar material, leaving it for a day or two, and then the spores will drop out and form this beautiful pattern there that you can see. And often the colour is important, but if you have a compound microscope, then you can look at the structure of the spores. For instance, some have particularly warty appearances, and they come in all different shapes. And if you have the software for that, you can also take measurements as well, which are also really important occasionally. For my literature suggestion for this one, I suggest Roger Phillips' Mushrooms. It's got fantastic colour photos of lots of species, many hundreds, not just the mushrooms, but also other fungi like crust fungi, subtle fungi, most of what you're going to come across. Now onto the vascular plants. So these are the larger, more flowering plants that you'll come across. Take photos of all the features that you can see. So that includes the flowers, the fruits, the stems, the leaves, everything really. Because some species that might have really similar flowers might have drastically different fruits, and vice versa, and all combinations on that theme. So take as many photos as you can. A hand lens can also be useful if you have one. These usually have a magnification of about 10 to 20 times, and they can ascertain the presence of glandular setae, for example, glandular hairs on the flower or the stem, which are often invisible to the naked eye, but can be made much clearer by a hand lens. As far as literature is concerned, the Bible for botanists is Stace, which is now in its 4th edition, entitled The New Flora of the British Isles. It's so comprehensive with loads of different, pretty much all of the wild species that you're going to encounter, lots of the naturalized ones and lots of the hybrids as well, with great keys and illustrations as well. But for beginners, then maybe take a look at the Collins Wildflower Guide, great illustrations and written by some really knowledgeable botanists. Now onto the mollusks. So this photo here is of one of my favorite ones, which I've never actually had a chance to see myself. This is the shelled slug. So you may think that a shelled slug is just a snail, but funny enough, there are shelled slugs that are not snails, but slugs. And this one has sort of a fingernail-like shell right at the end of its body, and is quite rarely seen and feeds on worms. But for some mollusks that aren't as easily identified, then it can be a good idea to check the mucus color, because for some genera like Arian or Arian, I'm not sure how to pronounce it, the mucus color can actually change between like orange and gray, those sorts of colors there. So check that if you can. Secondly, photograph the mouths. So these are obviously the mouths of the shells, not the actual snails, as that would probably be a bit more difficult. But with a smaller species like this, you can see by the scale bar that it's not the largest of snails. Then they all look rather similar, quite nondescript. If you look at the mouths, then they have specific patterns of teeth or spines. This one has one spine here at the sort of the base of the mouth, but they'll often have spines sort of around the lip, which differ between the species. For literature, I mainly use this fold-out guide on the left here. By the way, all of these are available on NHBS, all of the publications that I list. So if you're interested in getting any of them, this isn't actually a paid advert for NHBS, I just realized, and that's what it sounds like. But all these are available on NHBS, you can get them from there. On the left is a fold-out guide for snails, great illustrations. You can see on the bottom left here, one species that does have teeth on the mouth rim, and on the right, Slugs of Britain Island is a really good book as well. This is part of the Aid Gap series, which is aiming to cover a few more of the less studied groups. Now for arachnids, I've tried to choose the cutest photo that I can find. This is a jumping spider in the family Salticidae, And they don't really create webs to catch their prey, but instead they hunt on foot. And so they need large eyes on the front of their heads to spot and keep track of their prey. And this eye arrangement there is fairly distinctive for solsticids. And eye arrangement in general is really good for narrowing down the spider that you're looking at. Some spiders actually have 6 eyes instead of 8, which you may not expect, but that can also be distinctive for particular genera. But you don't have to actually have the spider with you to identify it all the time. Often the web structure can help to narrow it down as well, often to family or genus level. But here on the left is illustrated an example of one where you can actually identify it all the way to species level. This is the missing sector orb web weaver. And as you can see, it's missing a sector of its web, which identifies it as that species. For literature, my go-to is Britain's Spiders, a field guide. It's really good, plenty of great photos. Also illustrates a lot of the webs of different families, so you can narrow down your spider that way as well. A lot of arachnologists like the Spiders of Great Britain Island, which is split into 2 parts and is authored by Roberts. The first part includes the text and all the line illustrations, and the second part includes the color plates. Although it is extortionately expensive, which is a shame. But if you're really getting into your spiders, then that would be what I recommend. You've also got full scorpions in the UK, more commonly known as pseudo scorpions, which are absolutely tiny. They'll do no harm to you. They don't have a stinger, which is why they're pseudo scorpions. And they can be fairly common in leaf litter, but I'm always excited when I see them. And if you do, then this is the ID guide for you. Now on to springtails. Usually, as a rule of thumb, the larger the better, because a lot of the smaller ones require microscopy to identify. But this one, an Orthopteroid, doesn't. You can identify it by the patterns on the thorax and abdomen here. So try to stick with the larger ones until you get a microscope. But at that point, everything's fair game, and have fun with them. Secondly, pay attention to the antennal and abdominal segmentation. This can help to narrow your springtail down to a particular family or genus. So if you remember back to the tenothrix that I was talking about earlier in the introduction, that is a dichotomid springtail. And those are characterized by having the final antennal segment, which is the 4th antennal segment, much, much shorter than the 3rd one. Here is a catyanid springtail, and its 4th antennal segment is the longest segment. You can see it at the end here. In a dichotomid, this would be much shorter, so about that size there, if you can see my cursor. So that's just one example of how antennae can help, but also the abdominal segmentation. If I go back to the previous slide, you can see here that in this Orthopteroid, the 3rd abdominal segment here is the longest. In others, the 4th will be much longer than the 3rd. In some others, then all the abdominal segments will be the same length. So pay attention to that. For literature, unfortunately, there's not that much choice, but this one is pretty good. It's fairly comprehensive with a key to all the collembola which were confirmed in the UK at the time of publication. Since then, there have been a few new adventive undescribed species which have been found in the UK, including the one that I found new to Britain, which I mentioned at the start. So just be aware of that, but otherwise this is what I would recommend. Now onto the dragonflies and damselflies. This photo here shows a common clubtail. It's one of my favourite dragonfly species because it's quite rare. It's restricted to a few rivers in the south. This one was in West Sussex, and it's unique amongst British dragonflies by having a clubbed tail at the end there, which gives it its name. But my first tip is to notice any wing spots. This is a particularly good tip for the emerald damselflies, which are rather emerald green on the thorax and on the abdomen like this one. And those are most reliably identified by looking at the wing spots. This is a willow emerald, so a fairly recent colonizer to the UK, but it's spreading rather rapidly. It has pale wing spots at the end of the wings there. The more common emerald damselflies will have dark wing spots, while the southern emerald damselflies will have the wing spots half white and half black. Secondly, get both dorsal and lateral photos if you can. This is particularly good for the hawker dragonflies and the bluet damselflies, as they all look rather similar. This is an azure damselfly, and you mainly use the thoracic pattern for this. Well, I do at least. You can see the azure has a narrow pale blue band where the arrow is pointing on the top of the thorax here, with a thicker black band below it. In the common blue damselfly, this black band will be much narrower, with the blue band being thicker. In the variable damselfly, this blue band here is most often broken. So if you look at the damselfly from above, the thorax, it almost looks like two vampire fangs with blood dripping off it, which I think is really cool. For literature, I use Britain's Dragonflies. It's got great photos of all the common species, as well as migrants and vagrants, which show up occasionally in the UK. Now onto the Orthopteroids. So this is quite a large group in terms of the number of orders that it encompasses. It encompasses the crickets and grasshoppers, which form the order Orthoptera, also the Dermaptera, which is the earwigs, as well as the mantids and stick insects, but there's not that many of them in the UK. No native ones at least. This photo here is a wart biter cricket, a really large species and quite rare, so you probably won't have to identify it unless you're looking for it specifically. And it gets its name because allegedly, a long time ago in Sweden, people used to use this cricket to bite off the warts on people's skins because of its such strong jaws, which is a cool tale if it's true. Firstly, you can identify them by ear. If you're able to hear grasshoppers, then different species have different stridulations, so they're different songs. And if you look on the Orthoptera Recording Scheme website, they have species accounts with recordings of all the different stridulations of grasshoppers. And if you're able to see them, then look at the pronotal pattern. So the pronotum is the plate that is above the thorax, so where the wings are originating, it's just below the two. And it has two pale lines, most often in grasshopper species. You can see in this field grasshopper, these lines are sharply angled inwardly, around just above halfway. But in this lesser marsh grasshopper, they're subparallel, so they're almost straight, heading down the body. The most established guide at the moment, which I recommend, is the Photographic Guide to Grasshoppers and Crickets of Britain and Ireland. There's a new book coming out in the summer though, called Grasshoppers of Britain and Western Europe, which also includes the crickets and other related insects. I haven't looked inside it yet, but it looks pretty good and will include species that could make it over to the UK in the future. Now onto the Hermitsra, which are the true bugs. And my first tip for these is to stay away from the nymphs, because the nymphs, which are an earlier developmental stage, so after the egg will hatch into the first instar nymph, and then it'll move through different instars, and then you'll have the adult. And the adults can often look rather different to the nymphs. And so if you're comparing a photo of a nymph, for example, to a photo of an adult, you may think that they're different species, but they actually are. You can check whether your specimen is a nymph by looking for the presence of a wing membrane. You can see on the right, at the end of the abdomen, and you can see the membrane of the wing poking out there. It's quite dark in this shield bug, but it's not always that dark, but it is always there in adults. Secondly, pay attention to the patterning on the vertex, pronotum, and scutellum. The vertex is the part of the face between the two eyes. You can see the eye, one eye here and one eye on the other side, and the vertex here has two oval-shaped dark spots. The pronotum, again, is the plate that lies above the thorax. That's this sort of shield-shaped plate just behind the head, and it has two dark spots at the front and sort of trapezoid-shaped spots on either side, further back as well. And then the scutellum is this triangular plate between the wing bases, which also has two triangular spots there. And in leafhoppers like this Eupteryx, often the wing patterning is quite variable or quite similar between species. And so you most often need to refer to the patterning on the vertex and the pronotum and the scutellum to identify it accurately. Thirdly, my literature recommendation isn't actually a print book. It's this fantastic website by Tristan Bantock. It's still in progress, but it's really, really good for most families. You can scroll through species accounts in the gallery, which also gives information on similar species as well, which may not yet be incorporated into the gallery. So a really great resource there. Now onto the Hymenoptera. This is the correct photo that I've included in this slide, because on this spider here is the larva of an acrodactyla parasitic wasp. And I reared this particular wasp larva through into adulthood. What it does, the adult wasp will lay the egg onto the abdomen. The wasp will develop as a larva, sucking on the juices within the abdomen. And soon enough will control the mind of the spider somehow. I don't know how it does it, but it controls the mind of the spider to create a silk triangle almost, where the wasp larva can then hop off the spider, having played its mind tricks, and then develop some sort of a cocoon where it develops into adulthood. If I was presented with the adult wasp, it's an ichneumonids, I would have no idea where to start, because a lot of the ichneumonids look absolutely identical. They're fiendishly difficult to identify. But with this host data, I know that this parasitized Tetragnatha spider here, that already narrows it down to just one genus of wasps, which helps tremendously. So if you're able to, and you're keen on identifying ichneumonids, then try rearing them through, because that's the easiest way to narrow it down quickly. In a similar vein, maybe stick to aculeates. So aculeates are the bees, the ants, the solitary wasps that aren't parasitic, and the social wasps. This here is one of the solitary non-parasitic wasps, which will, it's a bee wolf. It will hunt down bees that are larger than itself, bring them back into nest burrows, where it will lay eggs, and the larvae will then develop on the bee corpses. Those are much easier to identify than ichneumonids and other parasitic wasps. For my literature recommendations, for bees, I'd go with the Field Guide to the Bees of Great Britain and Ireland by Stephen Falk. Fantastic illustrations by Richard Lewington. It has photos as well, so you can see what the bees will look like in the field. For ants, I like the book creatively entitled Ants by Skinner and Allen. It's quite comprehensive, lots of the species that you'll ever find in the UK, including some hothouse species as well. So those are species that aren't established outdoors in the UK, but have been introduced into the UK, into places like the Eden Project, where they can survive within these temperature-controlled environments. For solitary wasps, I like the book Solitary Wasps. Like the Ants Guide, it includes great keys, great illustrations as well. For ichneumonids wasps, because there's so many, there's not just one book that covers the identification of them all, but there is this Royal Entomological Society handbook, which acts as a sort of directory. Once you've identified it to subfamily or genus level, you can look in this book, and that will direct you to specific scientific papers that deal with the identification of those wasps. Now, if you're criminally insane, then you might want to identify chalcidoid wasps. And this is what you might be looking for. This is the keys to the insects of the European part of the USSR. A specific volume and part of that deals with the chalcidoid wasps. I just call it Medvedev because it's edited by Medvedev, even though Medvedev doesn't actually author any of the parts in this book. But it does key out all of the chalcidoid wasps that were recorded in the USSR at the time of publication, and it's the best sort of guide that covers them all. Now onto the beetles. So in a similar vein to the springtails, generally the bigger the better. Because there's so many of them, a lot of the rove beetles and the leaf beetles, quite tiny things, often need dissection to identify, which is not ideal for the casual naturalist. But larger things like this longhorn beetle, so called by the long antennae, which is so long I couldn't even fit it on the PowerPoint slide, are much easier to identify. This is the catchy named Agapanthia villosa viridescens. It's one of the more common longhorn beetles and quite recognizable too. Secondly, note food plant in the case of weevils and leaf beetles, like this alder leaf beetle, which feeds on alder almost exclusively. And so if you find something like this that's on an alder leaf, this is unhelpfully posing on what looks like a nettle leaf, probably fell onto there. And if you find it on an older leaf and it looks like this, then it's likely to be an older leaf beetle. And the same is true for quite a lot of weevils and other leaf beetles. For literature, a lot of it, because the Coleoptera is such a large group, a lot of the identification works are ordered by family. So you need to identify your specimens to family level first. And for that, I often use UK Beetle Recording website, which has a page entitled Beetle Families, where you can scroll through all the beetle families that have been recorded in the UK, and there are galleries of certain representatives from them. So you can find the ones that look most similar and then move to the family-specific identification resources. I found that easier than keying them. The Beetles of Britain and Ireland series is pretty good. It comes in 4 volumes, which cover all of the families, although one of the volumes, I think it's the 3rd volume, isn't yet published. It's pretty good, the good quality keys, but it is rather expensive to get all volumes of it. The Carabidae of Britain and Ireland is an example of an in-print Royal Entomological Society handbook, which I use quite regularly. That's really good for the ground beetles. There's a new book on the left here, the Hysteridae, Spheritidae and Sylphidae of Britain and Ireland by Lane Lucas and Ashley Whiffen. And this is excellent because it's not just an identification guide, but it's also primarily an atlas as well. So it has information on the distributions with distribution maps of different species. Probably the most recognizable beetles are the ladybirds. And for this, I use the field guide to the ladybirds of Britain and Ireland, again with fantastic illustrations by Richard Lewington, which also include the larval stages. So you don't just have to stick to adults for this one. But going back to Mike's insect keys, this is honestly my favorite website because beetle-wise, it includes pretty much all of the beetle families that you'll ever find with great keys to them. So you don't need to pay hundreds of pounds for books when all the keys are here. Now on to the flies, the true flies, Diptera. I spoke a bit about leaf mines earlier. These can often be easier to identify than adults. As the leaf mining flies, they're usually quite small, black to the naked eye, and so not that easy to identify. But if you have the leaf mines, those are much easier because they have specific shapes, patterns, where they originate in the leaf can be distinctive as well. And to identify those, and there's a few different websites that you can use. The one that I use most frequently is the ukflymines.co.uk. So that's a great resource for those. Secondly, wing venation can be key as well. So as I said earlier, wing venation is often quite invariable within the species, but between different species and different genera and different families of flies, it can actually vary quite significantly. So if you can get a clear photo of that, as that can help a lot in the identification process. For literature, like beetles, this is often spread out between different families. Most of the books cover the more sort of charismatic fly families. So you've got the British swordflies and their allies on the left, Britain's hoverflies on the right. Both are really good, fantastic photos and illustrations too. For crane flies, this is what I use most often. It's quite difficult to find. So if you go onto the catalogue of the crane flies of the world website, ccw.naturalist.nl, and then you go to the literature tab, then in the search field, type in Stubbs, S-T-U-B-B-S, go to the last page of results and then scroll down a little bit and you'll find keys to the families of crane flies and then keys to the species within those families. So that's a fantastic resource, and there's rarely a crane fly that I can't identify using those keys, except for Molophyllus. I'm not sure that anybody can identify those. Now on to butterflies. The habitat and the habits can be relatively helpful if you've got 2 butterflies, if you're comparing a butterfly to photos, because this is a black hair streak in this example, and they're rarely found away from blackthorn hedges, which is their food plant. Sometimes going a little bit further afield to find nectar, but rarely far. Whereas its most similar species, the white letter hair streak, feeds on elder in its larval stage. So it's most often seen flitting about the canopy of elder leaves, of elder trees. occasionally also coming down to ground level to feed. But depending on the flora around you, that can be helpful for identification. Also beware of worn individuals. So this is a long-tailed blue butterfly. It's usually, if it's in good condition and it's a male, then its wings will be totally blue. But this one is a migrant species, and this particular individual may have flown across the channel itself. And as would be expected, it's pretty worn, so it's pretty damaged. And this can change its appearance quite drastically to what you may be comparing it to. So do be aware of that. Book-wise, I would recommend the Britain's Butterflies book. Great photos in there, and again, very comprehensive. For moths, don't just stick to the adults, but the larvae also can be good ways to record a species presence. Sometimes the larvae can be easier to find than the adults. So a good way of creating biological records. Secondly, try to identify the moth while you still have it. So this pictured here is an elephant hawk moth. It's fairly distinctive and identifiable, but say it was a copper underwing and you photographed it, just the top side, released it, got home, and then you tried to identify it, and you realize that there's two very similar species, the copper underwing and the Svensson's copper underwing. You need to look at the palps. That's one way. You need to look at the palps to identify it, because they're different colors depending on which species it is, or the underside of the hind wing. And now that the moth has been released, you let out a scream of anguish as you realize that you just can't identify this particular specimen. So if you can, try to identify the moth while you still have it. I mostly use the Concise Guide to the Moths of Great Britain and Ireland for the larger macro moths. I like it because there's not too much text in it, it's mostly illustrations. And so when I'm at my moth trap and I'm trying to identify a particular moth, I can quite quickly flick through it. But also it's spiral bound. So if I'm at my moth trap, then I can also leave it flat and it doesn't sort of flip back closed again. So that's quite convenient for me. For micromoths, I use the book on the right, Field Guide to the Micromoths of Great Britain and Ireland. Again, great illustrations, but micromoths are a bit harder. This is the best treatment of them that I've seen so far. And for caterpillars, this is a book that my straw dot larva was posing on a few slides earlier. This makes caterpillar identification so much easier than it used to be. I used to really struggle, but as soon as this came out, my life got a whole lot easier because of the quality of the illustrations within. Now I'm so close to finishing now because there's so many taxonomic groups to get through. Now it's just the vertebrates. There's not much in the way of tips that I can give for these. These are my literature recommendations. For mammals, you've got the Britain's Mammals on the right there. There's a new edition, I think published sometime in the summer, June or August, I think, with really great quality photos in there. And for birds, I like the Collins Bird Guide because it also has an app version. So you can download it onto your app, onto your phone, and it takes up no space at all. You can take it into the field, and it also has different bird calls and bird songs loaded onto it as well, which you can play into your ear if you hear a bird sound that you don't recognize. So in conclusion, try to take photos from all angles because you're not sure what you might need to identify your specimen later on. Note the food plant or the habitat because it can all come in handy later when you're trying to compare two very similar species which may have different habitats or different food plants. Make the most of online resources because they're totally free. NatureSpot is a great way of just casually scrolling through the options, trying to narrow it down that way. The experts are there to help, so do bug them. They wouldn't proclaim their expertise if they didn't want you to use it, so do get in contact with them. And if you have any further questions or ID queries, you're not sure where to direct specific queries as well, then you can contact me on Twitter and I'll try to help as much as I can or direct your question to someone who's more knowledgeable than I am. On Twitter, I'm @my_wild_life. So do get in touch there if you have any questions. But that's the end of my presentation. So I'm happy to take any questions. And if there's anything that people need elaborating, if there's anything technical in there they want me to explain, then do pop it in the chat and I can answer that.
Wow, that was absolutely fascinating, incredibly comprehensive, and also just so interesting to hear about some of the specific kind of tests or little tips you have for when you're in the field or back home, especially that lichen one. I found that really interesting, like testing what the lichen is growing on helps narrow it down. Yeah,
that's cool.
Really, really interesting. We've had a load of great questions, so I'm just going to go straight in with the first one from Bev. She's asked, this sounds like a fair bit of dedication. Yeah. Where did you start out, James?
I think once I sort of got on iSpot and sort of testing all the different taxonomy groups there, I try to identify as much as I can in the field. And I start off with the easy groups, but as I sort of progressed, I would look for more challenges. So I started going on trying to identify beetles and then to springtails, which is probably the pinnacle of what I've tried to identify so far. But I mostly started off with butterflies and the bees, sort of the larger stuff that I didn't need a microscope to try to identify. So I think moths are also a good place to start because of the literature that's available for them. It's all quite up to date, doesn't require keys, and usually stays still longer than stuff like bees. So yeah, if you're looking for a suggestion of where to start, I'd say moths.
And there's a lot of moths, so you could be doing that and learning as you go for quite a while. I think that's a good recommendation. Now I know other James on this live stream. What did you start with?
Moths and butterflies. Yeah.
Okay.
Yeah, as a kid, loved butterflies in the garden. Went to a moth trapping when I was 12 or so, and was just hooked ever since. And then later on in life, I learned about birds and things, but birds, boring. But absolutely hooked on moths. Still am. And you can spend probably an entire life, going out trapping with migrant moths as well, with changes in distribution. I've casually trapping in gardens where I've lived, I've found records that are like the 10th for the county or the 1st for the county, or in one case, 3rd for Britain and things like that. So it's amazing just what effort a one group can yield. Anyway, this is James's talk. I'm going to stop talking about moths now.
Really interesting to hear that as a starting point. Here's another question from Gwen. This is a bit of a longer one. So I once went to a talk about lichens, and the speaker said that the distribution map for some species was more a map of where recorders lived than where the lichen was restricted to. Would you say this was true for a lot of groups or species?
It is. When you look at distribution maps on the NBN Atlas, for example, that's a really great source of distribution maps. You'll often find that they're clustered around maybe built-up areas, not because they exist better there, but because there's a higher concentration of recorders there. And this does sort of muddy the waters when you're trying to probably establish where the main hotspots for these species are. And especially when you're looking at taxonomic groups where the distribution maps are changing genuinely because of, say, climatic differences. For lichens, for example, the distribution of those has changed as pollution has decreased, where industry has been outsourced to other countries. The amount of pollution being emitted by these power stations has decreased. And this has allowed many intolerant species to recolonize into urban areas and places where they were previously completely absent. So those sort of biases created by where recorders are focusing on does sort of create problems there. So the more naturalists we can have focusing on all groups, the better.
Yeah, that's really, really interesting. Yeah. Okay, here's another one. Now, you mentioned some bits of kit sort of throughout the talk, but maybe you can itemize a sort of starter kit list, because there's a question from Lisa here. What would you say are the most important bits of kit to get someone started with identifying everything?
I would say that what I mostly use are my microscopes. I've got a stereo microscope and a compound microscope. The stereo microscope allows greater detail for sort of the larger invertebrates, whereas my compound microscope allows me to identify springtails better and algae and fungi. But if you don't really have the budget for that, then I'd get something like a sweep net that just allows you to find as much as possible, because the more that you're trying to identify, the more successful identifications you will make. So something like a swoop net, which you use to run through the vegetation, sort of a figure of 8 movement, can collect a lot of invertebrates and a lot of things that you would never otherwise see. And amongst those will be really distinctive things, which are much easier to identify. So I think anything you can get that increases the number of specimens that you are able to find is good. Another thing that you don't even have to buy, if you have a white sheet, you can place that under a branch, for example, get a long stick and whack the branch, and then invertebrates will fall from the branch onto this white sheet and become much easier to see. And then you can sort of return them to the branch once you've looked for distinctive things to identify. So anything like that, really.
Yeah, no, I like that. Like, find as much as you can, and then each success you have, there'll be more of them, even though, you know, you probably have to ignore 80% of what you found. Yeah, no, that's good. I like that. You start having fun straight away. Here's another question from Becca. Does it matter if you only have an hour every few weeks? I'm worried my progress will be slow and demoralizing.
No, I don't think it does at all. I think that if you keep chipping away at it every few weeks, then that's enough regularity with which you can still have a support, sort of a base from which to build on every single time. I think a problem faced by all naturalists, no matter how much time they have, is a sort of empty space left in the winter for flying insects where they aren't as active. And a lot of people I know, including myself, have to relearn how to identify bees and stuff every spring. So you wouldn't be alone in that regard. Everybody has to do it. So no, I think just an hour every week is enough to make a base and make some really important contributions to science as well.
Nice.
Can I jump in with one, Jack?
Of course, yeah.
Thank you very much. I've got a personal one. And James, some people might assume that a narrow focus on one taxonomic group, be it beetles or vascular plants, whatever, would actually be more valuable than having like a broad range of knowledge of a lot of different groups. Would you say that broad knowledge can actually be really useful and really, how does that manifest?
I think that there's definitely benefits to both approaches, both focusing on certain taxonomic groups and trying to sort of spread yourself as much as possible over a broader range. What I managed to do is focus on a taxonomic group that isn't much studied. So with Springtails, I'm able to sort of focus on them slightly and make contributions to those that actually are quite important, like new species to Britain and new species to certain counties and new species in their entirety. But I'm still able to maintain a base of all sort of taxonomic groups. So some sort of hybrid system is good, especially with taxonomic groups like Springtails, which are so understudied that you don't actually need huge amounts of knowledge to make significant contributions to them. I know that some naturalists, I think Graham Lyons said this once, he feels like not a lot of butter thinly spread over a slice of toast when it comes to all the broad range of taxonomic groups that he focuses on, and he does so incredibly well. But he still managed to make contributions to all of these different taxonomic groups. So I'd say some sort of hybrid approach between the two is best. But it's whatever you want to do. Both of those approaches, whether you spread yourself equally or you focus on a particular one, will still contribute to conservation via the data that you produce. So whatever you want, really.
What do you think is the most understudied thing?
I think that I might be biased and say that Springtails are up there. I'm often talking to people, including naturalists, and they've never heard of Springtails. It's a fairly common thing, and I can understand it totally. But also the algae as well, single-celled algae. There's so many species of them. In fact, let me get something from my bookcase. I was once gifted this book here, and it's a massive volume, and it costs several £100s. And this is what people use to identify single-celled freshwater algae. And it's absolutely massive, and plenty of species per page, and there's hundreds of pages, large pages. And this is much more than you'll ever get for springtails. So yeah, I think freshwater algae are up there. So if someone wants to focus on those and contribute to that, then I'll probably be grateful for that as well.
That's amazing. That's a real tome. We've had a couple of questions about sources, and you've listed some really good ones actually. But they're asking, how do you work out or find where the best sources are?
Okay. Usually it's just a case of speaking to experts and asking what they recommend. That's how I find most of those. Because as you start working your way through different taxonomy groups, and you'll sort of get in touch with experts either to confirm records, and you can ask what literature is best to confirm a certain species. And it's just stuff, what I've recommended is stuff that I've sort of collected over many years, but also just a search on a book retailer like NHBS. Other retailers are available, but searching for particular taxonomy groups on there will reveal the sort of breadth of literature that is available.
Right. Couple more good questions here. So one's actually asking how you knew when you were looking at the springtail, how you knew that it was a thorax segment and on abdomen.
So this does take a bit of learning. This is mainly through experience of trying to key out these springtails, because at the front of that FSC aid gap guide, there are illustrations to the morphological anatomy of these springtails. So once you get a particular piece of literature, they'll always have a section at the front that introduces the sort of terms that you'll encounter with loads of handy diagrams. So yeah, once you start to focus on groups like that, there's plenty of resources that will help you with specific morphological features. Right.
Yeah. Yeah. Here's a kind of more general question, which you touched on, but it'd be good to sort of know a bit more about. How do you work out what family it's in or what group it's in from the start? So if you're looking at something you've never seen before, how do you know that it's a ground beetle or it's a different kind of species or group? Or
yeah, that's a very good question. What I use most frequently for this is the NatureSpot website, because you can click onto quite broad taxonomy groups like beetles, for example, because you're usually fairly able to identify a beetle no matter what sort of experience you have. So go onto the beetles page, and there's quite a lot of pages of beetles because there's so many of them, but just scrolling through those to find the ones that fit best will get you started there. And once you do that more often, then you'll sort of gain experience and you'll be able to recognize them much more easily. And that does come quicker than you may expect. So before long, you'll look at something and instantly know that it's a ground beetle, for example, or a chrysomyelid beetle. Pretty much, yeah.
Yeah. Okay. I've got one of my own questions. So theoretically, what do you feel is the most, like the number that you could reach of species in the UK that you could identify realistically?
I know that the top pan species lister has about 12,000 species at the last time I checked, which is remarkable, but he is a full-time ecologist. So every day for his job in the on-season, he's going out to sites and recording species that he sees, pretty much all of the species that you can possibly find, and then identifying them in the winter. And that obviously adds so many species that I'm not sure I'd ever be able to reach if I don't become a full-time ecologist myself. So I say, it's a tricky question, because as I add more species, obviously the rate of additions decreases. So I'd be quite happy if I'd be ecstatic if I was able to reach anything near 10,000. So that would be an ambitious target for me, a lifetime target, I'd say.
Yeah, it's a nice goal. Rachel's asked, how do you find the time? Do you sleep? Do you find anything at night, actually? That's kind of quite a good question.
Yeah, well, while I'm sleeping, I have to use that to my advantage somehow, so I'll set the moth trap, and then I can catch things while I'm asleep. So that's how I manage that problem. I do sleep, yeah, sometimes not until quite late when I'm at my microscope. But yeah, usually I try to make the most of my after school hours on the weekends, but not too much, because I don't want to burn myself out too much. So it's important to have breaks. but use those to your advantage and find more things to identify later.
Yeah, take your time, go slow, enjoy it, I think.
Absolutely.
Yeah.
I've got another one, Jack, if you don't mind.
Okay, great. Yeah.
I want to know the most unusual place that you've ever suddenly come across an interesting lifer.
Well, the first one that comes to mind now is, well, I've got two that I've thought of. One of them, my parents won't be so happy for me speaking about on YouTube, but in my airing cupboard, which is just there, I actually put a Longworth trap, which is a small mammal, a humane trap for small mammals. And I found a yellow-necked mouse in there, which is sort of the scarcer cousin of the wood mouse that most people find. And I was quite ecstatic about that. My parents were less pleased, but that's one of the more unusual places. I remember also finding a spider that I'd long wanted to find in my geography classroom. I'm not sure where it exactly came from because I put my geography folder on the desk and a spitting spider suddenly ran out of it onto the desk, and I was amazed. None of my classmates quite shared my enthusiasm for it. It's such a cool spider with quite a humped thorax, and it will spit parts of web, I think, onto its prey to entrap them. That was a long-term target of mine that I finally saw in my geography classroom, so that was probably quite weird.
Oh, they're excellent. And what I like about those is my first and only yellow-necked mouse was caught under a colleague's desk at the RSPB headquarters. So, it's the scarcer mouse. It seems to really like coming inside buildings and getting into weird places. Yeah. In fact, my first spitting spider, I went out with a friend to look for the garden centre spider, the one with the fluffy legs. So we went and looked at that, and then I came home and I was like, great, I've seen a cool spider. Came home, spitting spider on the wall. So I got a nice little spider duo that day.
Brilliant. I want to see one of those greenhouse spiders. It's Euloborus plumipes.
That's the one, yeah.
Yeah, they've got such cool little feathery legs. It's quite awesome.
Yeah, well, and as you were saying earlier about going out to known sites for things to kind of get your eye in, the first time I ever saw it, I heard there was a location for it, went and saw it, got my eye in, and I've since found it at 3 other locations where I didn't know it was going to be. So advocating that approach of sometimes it's good to get your eye in and know what you're looking for.
Absolutely, yeah.
Yeah, that's amazing. I remember the yellow net mouse day. Yeah.
Yeah, it was under John's desk, wasn't it?
Yeah, it's fun. I miss the office actually, because often at the RSPB there'd be something on the window that someone would show you, or some unusual moth in the archway on the way in or something. Yeah, it's good times.
Yeah. Were you there that day that enormous tanner beetle landed on a window? Yeah, that's fantastic.
That was incredible. Absolutely incredible. Yeah. Right, that is all of our questions, and it's 4:15 now, so we're going to wrap up. So thanks very much for everyone who's attended. Really hope you enjoyed it. Huge thanks, obviously, to James, who's given a fascinating talk on identifying species that has really kind of blown my mind a bit. We're back from the brink. We've got a series of talks still to come. Some are on art and nature. One of them's on how to get green and be more environmentally friendly. I'll leave a link to all of our talks that are coming up in the chat just before we leave. And other than that, it's goodbye from me.
Goodbye from me.
And it's goodbye from me.
Great. Thanks, everyone. Thanks for coming, and hopefully see you soon at another talk.
Thank you.